You must be tired till now. But we still have two important organelles to cover. Let us talk about the synthetic functions of the Golgi apparatus. Well, a major function of the Golgi apparatus is to provide additional processing of substances already formed in the endoplasmic reticulum. It can also synthesize certain carbohydrates that cannot be formed in the endoplasmic reticulum. This is especially for the formation of large saccharide polymers bound with small amounts of protein, which are hyaluronic acid and chondroitin sulfate. A few of the many functions of hyaluronic acid and chondroitin sulfate in the body are that they are the major components of proteoglycans secreted in mucus and other glandular secretions. They are the major components of the ground substance, or non-fibrous components of the extracellular matrix, outside the cells in the interstitial spaces, which act as fillers between collagen fibers and cells. They are principal components of the organic matrix in both cartilage and bone. And lastly, they are important in many cell activities, including migration and proliferation. Processing of endoplasmic secretions by the Golgi apparatus, formation of vesicles. As substances are formed in the endoplasmic reticulum, especially proteins, they are transported through the tubules toward portions of the smooth endoplasmic reticulum that lie nearest to the Golgi apparatus. At this point, transport vesicles composed of small envelopes of smooth endoplasmic reticulum continually break away and diffuse to the deepest layer of the Golgi apparatus. Inside these vesicles are synthesized proteins and other products from the endoplasmic reticulum. The transport vesicles instantly fuse with the Golgi apparatus and empty their contained substances into the vesicular spaces of the Golgi apparatus. Here, additional carbohydrate moieties are added to the secretions. Also, an important function of the Golgi apparatus is to compact the endoplasmic reticular secretions into highly concentrated packets. As the secretions pass toward the outermost layers of the Golgi apparatus, the compaction and processing proceeds. Finally, both small and large vesicles continually break away from the Golgi apparatus, carrying with them the compacted secretory substances and diffusing them throughout the cell. Let us understand this with the help of an example. When a glandular cell is bathed in amino acids, newly formed protein molecules can be detected in the granular endoplasmic reticulum within 3 to 5 minutes. Within 20 minutes, newly formed proteins are already present in the Golgi apparatus, and, within 1 to 2 hours, the proteins are secreted from the surface of the cell. Two types of vesicles are formed by the Golgi apparatus: secretory vesicles and lysosomes. In a highly secretory cell, the vesicles formed by the Golgi apparatus are mainly secretory vesicles containing proteins that are secreted through the surface of the cell membrane. These secretory vesicles first diffuse to the cell membrane and then fuse with it and empty their substances to the exterior by the mechanism called exocytosis. Exocytosis, in most cases, is stimulated by the entry of calcium ions into the cell. Calcium ions interact with the vesicular membrane and cause its fusion with the cell membrane, followed by exocytosis, opening of the membrane's outer surface, and extrusion of its contents outside the cell. Some vesicles, however, are destined for intracellular use. When the cell membrane loses much of its substance every time it forms a phagocytic or pinocytotic vesicle, and the vesicular membranes of the Golgi apparatus continually replenish the cell membrane. In short, the membranous system of the endoplasmic reticulum and Golgi apparatus are highly metabolic and capable of forming new intracellular structures and secretory substances to be extruded from the cell.
Explore our extensive library of over 1,800 video lectures, to learn about a wide range of topics. Only on sqadia.com.